Hi guys! In this podcast, we will be covering the first 6 specification points in 3.11 monomers and polymers. Okay, so the first thing that is important for all of us to know is that although there is a lot of variety between and within different species in terms of their genotype, which are the genes responsible for certain traits, and thus their phenotypes, the expression of these traits, the biochemical basis for these organisms are still very similar in all living organisms. For instance, they all contain carbon-based compounds, such as nucleic acids. And this is important because it can be used to support the theory of evolution, as sharing biochemistry provides evidence of a common ancestor and thus indirectly supports the theory. Monomers are small units, and larger molecules can be made from these. In A-level biology, an example of this would be a monosaccharide. A polymer is formed when many of these monomers are joined together. An example of this is a carbohydrate. One way in which a carbohydrate can be formed is by joining a large chain of monosaccharides together. Other examples of monomers include amino acids, which are used to make proteins, and nucleotides, which are used to make DNA. So, how do we make polymers? Many are made through condensation reactions. In this, a chemical bond between monomers is formed and a water molecule is released. But what happens when we want to break down polymers? Hydrolysis reactions occur as the chemical bond between monomers are broken down and a water molecule is added. You have now reached the end of this podcast. Thank you for listening and have a wonderful day.